All right, so good morning, everyone. Our topic for today is all about cell modification. Okay. So as an overview, we're going to focus on the definition of cell modification, the types of cell modification, and their examples and functions. Okay, so what is cell modification? So they are specialized or modifications reacquired by the cell after cell division that helps the cell in different beneficial ways. Okay, so a cell modification or cell specialization or differentiation is actually a process that occurs after cell division, where the newly formed cells are structurally modified so that they can perform their function efficiently and effectively. Okay, so we have three types of modification, and under these types are the examples that shows how important the cell modification is. All right. So there are three major types of cell junction. So we have what you called the adherence junctions, the desmosomes and hemidesmosomes, or the anchoring junctions, and we also have the gap junctions or the communicating junction, and the last one is the tight junction or the occluding junction. So subjunctions are also especially important in enabling communication between neighboring cells via specialized protein complexes called communicating or the gap junction. So cell junctions are also important in reducing stress placed upon cells. Sometimes, similar communication channels are known as plasmodesmata, and in fungi are called septal pores. Alright, so what is apical modification? It is found on the apical surface of the cell. So it is specialized to carry out functions or at interfaces. So it includes secretion, what else? Absorption, movement, and luminal contents. All right. So let's start with cilia and flagella. So cilia are usually short, hair-like structures that move in waves, while flagella are long whip-like structures and they are formed from microtubules. Okay. So cilia are membrane-covered extensions of the entire apical surface. So they live in waves, often moving a surface coat of mucus material, while flagella are also concerned with movement. Okay. So these are the actual photo of cilia and flagellum under a high-powered microscope. So as what you have observed, cilia and flagella are cell organelles that are structurally similar but different in the length and functions. So cilia are present in organisms such as paramecium, while flagella can be found in bacteria and sperm cells. So cilia are shorter and numerous than flagella. Cilia and flagella are the most common organelles for locomotion in unicellular organisms. Ayan. So organisms with cilia can move faster and more efficiently. Okay, so recent researchers have discovered that the white blood cells move in similar fashion. So when the body injures itself, the WBCs traverse the blood vessels with the help of cilia. So most prokaryotic and eukaryotic microorganisms have flagella, but they differ from each other structurally and functionally. Okay. So now let's move on with villi and microvilli. So villi are finger-like projections that arise from the epithelial layer in some organs. They help to increase surface area allowing for faster and more efficient absorption. Okay. So while microvilli are smaller projections that arise from the cell surface that also increase surface area, allowing faster and more efficient absorption. Okay. All right. Okay. So, ayan. So, the main difference between villi and microvilli is that villi are small projections on the mucous membrane. So, particularly in the small intestine. Ayan. So, but microvilli are tiny extensions that mainly occur on the cell membrane of different organs. Furthermore, villi increase the surface area when microvilli are responsible for absorption and secretion. So, increasing the surface area. Ayan. So, in brief, uh, villi and microvilli are two types of projections that occur on the internal surfaces of different organs. So, generally, both are responsible for increasing the surface area. Okay, so what is pseudopods? So it's temporarily irregular lobes formed by amoebas and some other eukaryotic cells. Ayan, they are bulged outward to move the cell or engulf prey. Okay, so pseudopods are actually extensions of the cytoplasm. So what is cytoplasm? They are the thick liquid inside organisms like amoeba. So the microorganisms can change the shape of the pseudopod, making it move, appear, and disappear. So the pseudopods are used in movements and as a tool to capture their prey. So in order to move using pseudopods, the organisms pushes cytoplasm towards one end of the cell, which makes a projection or a pseudopod of the cell. Okay? So, this projection holds the crater in place and the rest of the cell can follow. So, thus, excuse me, moving the organism forward. So for feeding, organism extend their pseudopods to engulf their prey. Okay? So let's move on with the ECM or the extracellular matrix. So it is a compound secreted by the cell on its apical surface. Cell wall is the extracellular structure in plants cells that distinguishes them from animal cells. So glycoprotein is the main ingredient of ECM in animal cell. Okay. So, all right. So, this is the ECM. So, a major component of the extracellular matrix is the protein collagen. Ayan. So, this is the actual structure of the ECM. As what you have observed, the collagen proteins are modified with carbohydrates. And once they release from the cell, assemble into long fibers called collagen fibrils. Okay. So, what is the main purpose of this collagen? Ayan. So, collagen plays a key role in giving tissue strength and structural integrity. So, in the extracellular matrix, collagen fibers are interwoven with a class of carbohydrate-bearing proteoglycans, so which may be attached to a long polysaccharide backbone as shown in your screen. So the extracellular matrix also contains many other types of proteins and carbohydrates. So the diagram showing the extracellular matrix and its connections to the cell. Ayan. Okay, so aside from this, a network of collagen fibers and proteoglycans is found outside of the cell. So the collagen connects to integrated proteins in the plasma membrane via fibronectin. So on the inside of the cell, the integrins link up the microfilaments of the cytoskeleton. Ayan. So the ECM is directly connected to the cell it surrounds. So some of the key connectors are proteins called integrins, which are embedded in the plasma membrane. So the proteins in the extracellular matrix, like the fibronectin molecules shown in green in the diagram or the illustration in your screen, can act as bridges between integrins and the other extracellular matrix proteins such as collagen. Okay, so in the inner inside of the membrane, the integrins are linked to the cytoskeleton. So the integrins anchor the cell to the extracellular matrix, and in addition, they help it sense its environment. They can detect both chemical and mechanical cues from the ECM and trigger signaling pathways in response. Okay, so for examples, blood clotting. Blood clotting provides another example of communication between cells and the ECM. So when the cells lining a blood vessel, when the cells lining a blood vessel are damaged, they display a protein, what do you call this? Protein receptor called the tissue factor. So when tissue factor binds to a molecule present in the ECM, it triggers a range of responses that reduce blood loss. So for instance, it causes platelets to stick to the wall of the damaged blood vessel and stimulates them to produce clotting factors. Okay? Alright, so let's move on with the basal modification. So they are found on the basal surface of the cell. Okay. Ayan. Ayan. So let's move on with desmosomes and hemidesmosomes. So what are they? Anchoring junction on the basal surface of the cell. They are rivet-like links between cytoskeleton and extracellular matrix components such as the basal lamina that underlie epithelia. And they are primarily composed of keratin, integrins, and cadirins. Ayan. Okay, so the key difference between desmosomes and hemidesmosomes is that the desmosomes is directly form the cell-to-cell adhesions, while the hemidesmosomes form adhesions between the cell basement membrane. Ayan. So when we say cell adhesions and cell junctions, it is important to maintain the integrity of particularly or particular tissue and for enabling the cell-to-cell signaling mechanisms. So there are different types of cellular adhesions in all species of higher level eukaryotes. And it is important to differentiate between different cellular adhesions to understand more about their biological mechanisms. All right. So your screen is hemidesmosomes, which is a type of cellular junction. Ayan. So they are small and stud-like structures, and primarily they are found in the epidermis of the skin. Okay, so most what do you call this? Keratinocytes keratinocytes contain hemidesmosomes in between them. So they are present in sites where the epidermis gets attached to the ECM. Thus, the hemidesmosome is attached to two surfaces simultaneously. So hemidesmosomes distribution is also seen in the epithelial cells. They form connections between the epithelial cells and the lamina lucida. Okay, so hemidesmosomes are also involved in cell signaling pathways. All right. So now let's move on with the lateral modification. So under this are tight junction, adhering, and gap junction. Okay, so first we have the tight junction. So they act as barriers that regulate the movement of waters and solutes between epithelial layers, and they prevent leakage of ECF. Okay, so tight junction are virtually, but also partly selectively impermeable seals that encircle cells and bind them together into leak-proof sheets. Okay, so in other words, the plasma membranes of adjacent cells essentially fuse together tightly in order to limit the leakage of various substances between the two cells. So when we say tight junction, they are located within our body's epithelia. So epithelium, we say epithelium is the plural form of epithelium. Epithelium is a word that refers to the covering of the body's internal and external surfaces. So, okay, so this includes organs like your skin, the blood vessels, and the cavities. So thus, these tight junctions serve various functions depending on what the epithelium is in question. So in the skin, they keep us somewhat watertight and help keep allergy or allergens of our body. So in digestive system, they help prevent the leakage of digestive enzymes into our bloodstream. And also, the tight junction serve as a structural support mechanism that help keep the epithelium together. All right. Okay, so a second one is the adhering junction. So anchoring junction on the lateral surface of the cell, they are very similar to the anchoring junction of the basal surface of the cell, and they fasten cells to one another. Okay, so basically adherence junctions are involved in a number of critical functions, including providing additional structural support. So for example, they hold cardiac muscle tightly together as the heart expands and contracts. Okay, so adherence junctions are built primarily from cadherence. So when we say cadherence, it is any of a class of transmembrane proteins important in maintaining tissue together. Okay? Ayan. So catenins are connected to actin filaments. So when we say catenin, they are any of a class of proteins that have a role in cell adhesion. All right. Okay, the last one is the last one is the gap junction. They are also known as communicating junctions and closable channels that connect the cytoplasm of adjoining animal cells. And they are presence of connexon that allow direct exchange of chemicals between the cytoplasms of the cell, of the two cells. All right. So, initially, the need for signaling is a function of a gap junctions that form pores connecting adjacent cells. So small molecules and electrical signals in one cell can pass through the gap junction to adjacent cells. So this process allows tissue to coordinate responses to stimuli. So for example, gap junctions permit coordinated movements of muscles leading to childbirth. Okay? Okay, so that's it for your attendance. Just comment below your name and your section and short key takeaway about our discussion. Thank you for listening and hope you learned a lot today. God bless and always remember that you are all amazing.